Okay, next speaker, please, is Claus Michael Lehr. There you go.
Yeah, thank you. It's okay. Good morning. Yeah, I guess I'm going to switch a little bit gears indeed towards that more therapeutic application of the genome editing. But before I come to that, I'll give you a little bit more an overview of the context in which we are doing that. For those who might know me, I'm still professor at Saarland University, which is the southwest of Germany, close to the French border. But like 5 or 6 years ago, I had the opportunity to join a larger organization, which is Helmholtz, and to start a new institute. And just last year, actually in October, we had the inaugural building. What we are doing is a little bit different to what's been mostly discussed at this meeting. We have a focus on infectious diseases. And why we do have that is actually due to the fact that there is quite a gap in the development of novel drugs. And on the other side, if you look at the prevalence and upcoming of bacterial resistances, it's quite a threat when you on the other side see that the number of new compounds and even the number of companies that find it worthwhile to work on infectious diseases is going down. So there is like a public need to do more, and that's when Helmholtz in Germany becomes active. And in particular, we have an approach that is related. Here you see again the relevance of these compounds. We have a mission to work mainly on natural products, bacteria-based agents. So some of my colleagues who are more on this discovery and actually look at compounds that, for instance, these myxobacteria can make, and they're quite clever in hitting and overcoming their bodies. That's kind of the overall context. And what we are doing in my department is the taxi driving, is the delivery to the site of action. And my personal approach over the years has been always that I wanted to use non-invasive approaches, so trying to avoid the needle whenever it is possible. So that means what you mostly prefer is to do give things orally, then you have somewhere to cross the intestinal mucosa. You can inhale things also to treat diseases of the lung, also with the perspective of a systemic delivery via the lung. I'm going to talk about this a little bit more in the context of this talk. We do have some activities on the skin, and being an institute dedicated to infection research, there's a whole bunch of novel biological barriers, even of some pharmacokinetic concepts when you think about intrabacterial pharmacokinetics, which unfortunately today I have no time to address. What we are doing can tactically be split in two things. The one side is what some people refer to as biopharmacy. I have a deep trust in the usefulness of cell and tissue-based in vitro models, preferentially when they're human-based, because then we can really shed light in the black boxes and see what's going on at the air-blood barrier, for instance. And when it's all human, we don't later on have the problem to transfer from mice to man. The other side, if I would be only doing this, I probably would be a biologist, but I'm a pharmaceutical technologist, and that's what brought me actually to that area. I want to see how our little, not always, but often nano robots and carriers are dealing with these tissues, and so we make some of them as well. So, talking about the lungs, we don't have time. It's a tremendous area for drug delivery for two excellent reasons. First is the large surface area. Total surface of the respiratory airways is like a tennis court. That little towel in the middle is just the surface area of the bronchi. It's not only the size, it's also the extreme thinness of this barrier. This is a red blood cell, typically like 2 by 6 micron. This is the alveolar epithelium, basal membrane, and endothelium. So you appreciate less than 1 micron of cellular barriers to cross over such a large surface area. It's what we are doing with each breath, with oxygen and CO2. Why not using that more for drug delivery as well? We found out, and this is for the people who are interested in the details, are invited to read this opinion paper where we're kind of proposing pulmonary delivery is actually nowadays going beyond aerosol science. We do have a fair control of inhaling things and make good particles and inhaler and things. The critical thing is actually to get control on what happens after landing, so to speak. And there are these cellular barriers on the cellular end. In particular, I would like to bring the attention to the extremely thin cells that we have been seeing before. We have been working quite a while ago on making a primary cellular system which is human-based, and as you can see on the photo on the left side, this is really a tight epithelium with functional tight junctions, giving you, for illustration, epithelial resistance of 1,000 and 1,500 ohms per centimeter. This is a cell line which many people have been using, but as you can see, it's cancer-derived and therefore tight junctional barriers, fairly incomplete. In fact, it's leaky like a sieve. You could do a lot of useful studies, just absorption and permeability studies are of limited value. Primary culture is the thing to do. We have good connections to hospitals where cancer patients get rid of a whole lobe of the lung. We get a piece of tissue. This one was a heavy smoker. It's a cumbersome procedure. So luckily we succeeded, and the date the paper came just out, it was quite an ambitious project together with our colleagues to start an immortalization. And to make the long story short at that end, we now do have a cell line in hands which is human, which is alveolar type 1 characteristics, which grows to several passages and maintains the paracellular tightness. Switching gears again, on top of these cells, there's also something. Non-cellular barriers. And in fact, in the lungs, but also in other areas, we have two. One important one is mucus that covers all the mucosal epithelia. And in order to cross the mucus, apart from the fact that there is a dynamic barrier of mucociliary clearance, your nanoparticles have the same problem like these penguins who want to go to the sea. If the sea would be the area when they are in, would be like the cells, and that they can pass. But how to cross the mucus? This is seriously an issue. Other people compare the mucus with the Himalaya, and if you wanted, if the nanoparticle would be the size of a man, this is like you have to climb up to Himalaya mountains if you want to go to the top. So this is nothing trivial. And we thought it's worthwhile to study this a little bit more systematically. And there has been quite a bias in the literature about mobility of particles in that mucous barrier. Some people looked at short distances, looked at micromobility, and then you have to deal with the fact, that's what we found out from modeling and also from this is a cryo-EM study, that you have voids which are as large as 500 or up to 800 nanometers. You can imagine that in such a space, a nanoparticle can move and do Brownian motion quite comfortably, but it wants to pass through over a longer distance, going through all that building block of ice. It's a different story. So it's too much to go into the details, just to raise your interest. With this new paper that recently came out, we come to something, we are in Switzerland actually, it's the Swiss cheese model of mucus. Inside there are pores, but you can't see these pores. Like if you would never have cut the Swiss cheese, you would not see that there are holes inside. So outside is quite dense, and that's why when a particle lands, has in the first place even more difficulties to penetrate into the mucus and then finds way to see that. That's just a little bit the basic stuff that we are doing aside. On top of the mucus, there is still something else that is even in regions of the lungs where there's no mucus in the deep lung, the so-called surfactant. And as everybody is talking about corona in the context of bringing particles to plasma, we argued and raised the hypothesis, well, there are different proteins and different biological media in this lining fluid than the plasma. So most likely there will also be a different lung-specific corona around these particles. And the interesting point is that we do have lipids and some particular proteins that are different to those prevailing in the plasma. Also, if you want to see details, I refer you to that paper, and there is also a poster about this from Simon Resch outside. So what we do get is this coating, and that governs actually the fate. To summarize the major finding here is, regardless if you take quite different nanomaterials, hydrophilic ones or hydrophobic ones, the nature will always find the right primer of proteins that allows then in the second step the lipids to go on, and essentially it makes all the particles, regardless they're intrinsically different, quite similar. And now teleologically you can think what this all means, so that the lung clearance system acts like a snowplow. In toxicological sense, and that was it was made for, oh, wrong button. This is a good news, because whatever the material is, the lung is professionalized in handling it all and kicking it out. If you want to overcome it in purpose, in order to say, well, we want to go to a macrophage to treat malaria, or we want to go inside an epithelial cell to change some of the genes, what I'm going to now in a minute, we might have to look for some very complex rationalities. Okay, that was the fundamental, basic stuff that I have fun to do. And now come to the story where we show a little bit more about the application. And I was indeed approached by a colleague, Michael Korman, who is working on pulmonary diseases in the Children's Hospital. And actually, the disease that he is working with is a lung surfactant protein deficiency. There are 4 surfactant proteins. They are life important, otherwise the lung cannot be filled with gases. And there are babies, unfortunately, who are lacking the production, especially of surfactant protein B. The tragedy is actually they can never really breathe, so that means they are doomed to die soon after birth and cannot really be saved. In order to find a cure for that, these colleagues had actually developed a mouse model. Yes, occasionally I do also work with mice models because, you know, that's the thing in between. These mice are constructed in such a way that they have a deficiency of surfactant protein B that is corrected by a gene that has a doxycycline-dependent promoter. So whenever these animals get doxycycline, the gene is working, the surfactant is being produced, and the animals are fine. If you withdraw the antibiotic from the food, they're going soon to die because they don't make the surfactant anymore. If you now had a way to repair this gene and correct by editing the sick gene by a permanent promoter, then you could actually make these animals survive without giving them doxycycline. And that actually was the plan. So what we wanted to do, what Michael's proposes, was let's take this gene for the correct one that you enter here from the left side, and then you need one of these, we use the type of zinc finger instrument to cut the right part out, but because you can not really administer that protein into the cells, you take a messenger RNA that is encoding that nuclease. So what you do is, in a sense, a double gene therapy. First the plasmid and then the messenger RNA. And so if this works all well, the scissor is being produced by translation, it cuts out, and the rest, that is, I would say, almost history now. That is the typical genome editing part. For us was the clue if you would be able to do that, actually. So then the protein would be missing. What we have been working over the years was nanotechnologies based on biodegradable polymers, particularly one combination of chitosan and PLGA, which makes us positively charged particles that we can use for binding and complexing nucleates. We had been using plasmids, siRNA, so we thought this time maybe we can be using this messenger RNA approach by this technology. And well, you can see here from, this was a studies in Cell, that actually the protease that we wanted to encode was expressed, so that was a sign for good hopes, actually. And then we did the real experiment in their mouse model. So we started with an instillation of the adenovirus-associated plasmid, then waited for 7 days, gave the second gene therapy with the messenger RNA encoded system, and we used here a viral approach and we used a non-viral approach, and then we took the doxycycline away and we waited to see what the animals were doing. And that's the result of this study. All the controls actually died as expected within 3 to 5 days. Those treated with the messenger RNA survived significantly, and the interesting thing is actually, and I had rarely seen this before, that the non-viral nanoparticle carrier system is not only safer than the viral in a sense, also the numbers is a little bit small, so I would not dare to say this is really significant, but they even survived a little bit better, and so that's a good sign. If you would be translating that, we are far away from this actually. You would have to do it repeatedly, but let's say treating a patient every 30 days is probably a better approach than doing it, let's say, every day, or to die after 5 days. And I think in the interest of time and maybe some discussion, we will leave it with that. Thank you very much.
Going on? Yeah. Any questions from the floor?
Thank you. Do I understand correctly that the treated animals in the end also die, and if so, what do they die of?
Yeah, they die because these cells that are transfected, they are that for the typical lifespan, as long as these cells. What you need to do is you need to transfect type 2 cells, actually. The type 2 cells, I showed you before the type 1 cells. Type 1 cells are more the cells that are responsible for the barrier. Large in surface, small in number. The type 2 cells are those in between, which the main function is, let's keep it with this way for simplicity, is to produce this lung surfactant, the phospholipids and the surfactant proteins. So the genetic defect must be there. And what you do is, when you give this by inhalation in this model, you may be able, nobody knows how many, but apparently sufficient cells have been successfully edited in their genome that they have learned to produce this surfactant independent of doxycycline. However, when these cells die, and typically like 30 days is a normal life cycle for cells to die, then there is no cell left that could do that. So you would either have to repeat the thing, or what you really would perhaps like to consider is if you could expand that to a progenitor cell, so if you would be able to do the same thing with stem cells that you, let's say, have the system inherently, then perhaps you could really cure. Admittedly, this is far from really a real translation, but in a sense, I enjoy this, that it works so nicely from the delivery point of view and the technology that we were able to do that at that level. Now you have to take the next steps. Yeah.
Do you have a feeling for how many cells are being transduced, corrected in this model?
Honestly speaking, I don't. I mean, what I have learned from the people who have been working on cystic fibrosis, which is similar, but actually this is quite different, yeah, just that we understand each other. I mean, most people know this is the chloride channel, which you try to insert and to repair. This was the surfactant protein. That very few cells are actually transfected, but you must have enough. So enough to secrete some chloride, enough to lower the surface tension and to get functionality of the, so probably it's rather low. That means there's room to improve it.
It's intriguing that the microparticles are working better than the virus. Is this a particular site where the viruses are particularly bad, do you think? I mean, do you think, you know, non-viral methods would be better than viral methods for other applications?
This is a little bit of speculation, but apart from the fact that we can, let's say, manufacture these polymeric particles better than doing with the viruses, but that's maybe a question of the expertise. You also need, and this is not all sorted out in detail, you need also to aerosolize these systems. I mean, we are talking about the nanoscale and see an advantage that small nanoscale particles have an advantage to go into cells and to get trafficked inside the cells. However, when you want to have an aerosol delivery, you must be more in the micrometer size. So there's always, if you want to do efficient in vivo pulmonary delivery, you must do like both, having the nanoscale, small particles, but to carry them aerodynamically down, you need like a microtechnology. And I believe that the nanoparticles stabilize maybe better than viruses.
Very interesting. Thank you. Okay, can we have the next speaker, please? That's Umit Pool from